Good afternoon to all the EAPCET aspirants, and here we are starting a new chapter, that is histology and anatomy of flowering plants. From this chapter, in every year EAPCET exam, definitely there are 2 to 3 questions. If you consider all the EAPCET PYQs, definitely there are 2 questions from this chapter in every year. So, if you have to look into the contents of this chapter, like what you have to cover, this is a very broad chapter again, wherein you have to learn this chapter by fragmenting it into chunks again, okay, wherein first you have to finish off histology and anatomy of flowering plants. How you have to go through this chapter? This is a very broad chapter, you have to learn it point by point, segment by segment again. This chapter you need to fragment it into 3 parts. One is tissues, second will be tissue systems. In tissue systems, you will come across 3 tissue systems: epidermal tissue system, ground or fundamental tissue system, and vascular tissue system. Fine, right? Next, after tissue systems, you will come across the comparative analysis between dicot, monocot, root, stem and leaf. Fine. And lastly, secondary growth. Fine. So, under tissues, what you will be covering, beta? Both, based on the origin, based on the activity, you will cover first meristematic tissues and then permanent tissues. In meristematic tissues, primary meristematic tissues, secondary meristematic tissues again. Primary meristematic tissues based on location, you will learn apical meristems and intercalary meristems. Fine, right? This is the basic fragmentation of this chapter, anatomy of flowering plants. This is how you have to go through. First tissues and then tissue systems and then secondary growth. In today's class, we are going to cover completely about tissues, beta. We are going to finish off the tissues, okay. So, here you see, here we go. First of all, what is the actual definition of the tissue? What is the actual definition of a tissue? You can define a tissue as group of cells which are common in their function and they have common origin, but they can exhibit structural similarity or dissimilarity. Group of cells which can be structurally similar or dissimilar, but they have common origin and common function. Such group of cells are considered as tissue. Very important definition, ma. Take a note of it, like group of cells having common origin, common function, which are similar in structure or dissimilar in structure, which are similar or dissimilar in structure. But what should be common? Origin and function should be common. Structural similarity or dissimilarity can be exhibited. Okay, these are considered as what? Tissues. Okay. Now you have to know the classification of plant tissues in detail. Plant tissues primarily based on their function, if they have the ability to divide or not, based on that ability to whether divide or not, they are of 2 types, meristematic tissues and permanent tissues. Meristematic tissues are those tissues which divide continuously. They will divide continuously. Whereas permanent tissues, they stop the division and they will differentiate. Those tissues which stop division and differentiate are said to be the permanent tissues. So, here if you see meristematic tissues, again based on origin, they are of 2 types, primary meristems and secondary or lateral meristems. Primary meristems are helpful in the formation of primary plant body. Primary meristems are present from the beginning of plant life and they are helpful in formation of what, ma? Primary plant body. primary plant body. Secondary meristems will exhibit secondary growth and they play major role in increasing the girth or thickness of the plant. They will increase the girth or thickness of the plant. Here you see primary meristems, again based on location, they are of two types. Primary meristems based on location. Based on location, they are of two types. One is apical meristem, location or position also you can say. Based on location or position, they are of two types. One is apical meristem, second is intercalary meristems. Apical meristems are present at the apical regions of the plant, either at shoot apical meristem or at root apical meristem. These two are what beta? Apical meristems. They are at root apical meristem or shoot apical meristem. Fine, right? Shoot apical or root apical meristem. Next, intercalary meristems, they are present at the internodal regions in between two nodes. Intercalary meristems present at the internodal regions in between two nodes. This is all about primary meristematic tissues. If you see secondary meristems or lateral meristems, secondary meristems, they are known as secondary meristems because they play major role in the secondary growth. Lateral meristems because they appear in the later part of plant life. They are of two types based on their position or location again. If they are present in the vascular tissue or in the stellar region, that is vascular cambium. If the cambium is present in the extrastellar region or cortex region, it is known as cork cambium. Vascular cambium and cork cambium. Clear with this note beta? Vascular cambium again, if the cambium is present in between xylem and phloem tissue, that is considered as intravesicular cambium or vesicular vascular cambium. Vesicular vascular cambium. Fine. If at all it is present in between two vascular bundles, this is one xylem phloem and xylem phloem. If it is present in between two vascular bundles, it is considered as intervesicular cambium. One is intravesicular cambium or vesicular vascular cambium. It is present in between xylem and phloem. If cambium is present in between two vascular bundles, it is said as what? Intervesicular cambium. Till here it is clear, no ma? Once again, quick recap on meristematic tissues. Meristematic tissues are those tissues which will divide continuously based on their origin. They are of two types of meristematic tissues. One is primary meristematic tissue and the second is secondary or lateral meristematic tissues. Primary meristematic tissues, based on the position, we have two types: apical meristems and intercalary meristems. Apical meristems, it can be root apical meristem or shoot apical meristem. Intercalary meristems, they are present in between two nodal regions, that is in the internode. Secondary or lateral meristems, they take part in the secondary growth and they appear in the later part of plant life. Based on their position again, if it is present in the stellar region in between vascular bundles, it is addressed as vascular cambium. If the cambium is present in the cortex region, which is also known as extrastellar region, that is considered as what beta? Cork cambium. Here vascular cambium, again of two types: intravesicular cambium if it is in between xylem and phloem. It is addressed as intervesicular cambium if it is present in between two vascular bundles. Clear till here beta? Clear till here? Now we will see permanent tissues. Permanent tissues are those tissues which have stopped the activity of division and they got differentiated both structurally and functionally. Based on the types of cells that are present in the tissue, we have two types of permanent tissues. One is simple permanent tissue and the second is complex permanent tissues. Simple permanent tissues consists of those cells which are homogeneous. All cells present in this tissue are alike or same. All the cells present in the tissue are alike or same. Those are simple permanent tissues. Example: parenchyma, collenchyma and sclerenchyma, of which plant body is abundantly composed of parenchymatous tissue. Complex permanent tissue will have group of different types of tissues. are present in the complex permanent tissue. Example, xylem and phloem. If you considered in angiosperms, xylem consists of 4 different tissues which includes tracheids, vessels, xylem fibers and xylem parenchyma. Okay. Phloem again consists of 4 different types of tissues which includes sieve tubes, companion cells, phloem fibers and phloem parenchyma. Clear, right? Clear till here, ma? The differentiation of tissues. I hope this is clear. Now we will begin with the meristematic tissues. Before dealing with the meristematic tissues like apical meristems and intercalary meristems, it is very important to know the general characteristics of meristematic tissues. The general characteristics of meristematic tissues include they have the ability to divide, ability to grow and divide, and they are small immature cells. They are isodiametric. Isodiametric means they have equal dimensions in length, breadth, okay, in length, breadth and height. All the cells will have equal dimensions. They can exist in any shape, round, oval, spherical, polygonal, they can exist in any shape, but all the cells are isodiametric. They have equal diameters. Next, intercellular spaces are absent in between meristematic cells. And then all the cell walls, as they frequently divide, they do not have secondary cell wall depositions, they have thin cell wall. Remember, that is a simple logic. Meristematic cells means they divide frequently. They are dividing frequently means the cell wall will be very thin and parenchymatous. They are parenchymatous cells with thin cell wall which is cellulosic, thin, elastic and made up of cellulose. Okay. And in each meristematic tissue, nucleus is dense and conspicuous, cytoplasm is also dense. Vacuoles are completely absent or at least present. As they divide continuously, their respiration rate will be high. Rate of respiration will be very high in the meristematic tissues. So, quickly recap the characteristics features of the meristematic tissues. What are the characteristic features, beta? Firstly, you remember the function of the cells, then you can easily analyze the structure of the cells. The function of the meristematic cells is what? Division. Continuous division. Continuous division means intercellular spaces will not be there. Cell wall will be very thin, elastic, made up of cellulose. Cells will be small. Nucleus will be dense and cytoplasm is also dense. Vacuoles will be limited, primitive or absent, and rate of respiration will be very high. These are the keynote features of what? Meristematic tissues. Now, types of meristematic tissues. Before going to types of meristematic tissues, these are the 2 diagrams you have to have a keynote on it. This is extracted from root primordia and shoot primordia. This is root apical meristem, and shoot apical meristem. Most of the students will just by heart these labelings, but you do not do in that way. Here you first identify the meristematic zone. We all know from the peripheral diagram of morphology, from regions of root, that at the apex what is present, beta? Apex thimble like structure at the apex is what? Root cap. So, root cap is present at the apical region here. This is what? Root cap. Whereas meristematic zone is present proximal to the root cap region. Proximal or above to the root cap region, what is present? Meristematic region is present. This meristematic region has what? Root apical meristems. This is the hub of the meristematic cells. They have what? Root apical meristems. So, root apical meristem is present at subapical region. Remember, root apical meristem is present at subapical region or subterminal region. Take down, ma. It is present at subapical or subterminal region, whereas root cap is present at the apical region. Now, in close proximity to this, next to this, what do you have? Region of elongation and region of maturation. So, here what cells will be there? Cortex and central cylinder. So, close to this root apical meristem, what cells are present? Initials of cortex and central cylinder. Here you can identify at the subapical region, root apical meristem is present. Close to the root apical meristematic region, you have initials of central cylinder and cortex. Fine, right, ma? You have initials of central cylinder and cortex which will divide and they will form matured tissues, cortex and central cylinder at the region of maturation. Okay. Now the outer layer is called as what? Outer layer is protoderm, which will later mature into what? Epidermis. This will later mature into epidermis. Okay. Once a quick recap on this, at the apex region you have root cap, subapical region or subterminal region you have root apical meristem. Close to the root apical meristem you have initials of central cylinder and cortex. At the maturation region, at the region of maturation you have cortex and central cylinder. The outer layer here is called as protoderm. Protoderm will be matured to epidermis. Clear, right? Clear with the root apical meristem diagram. Now you see shoot apical meristem. Unlike root apical meristem, shoot apical meristem is present at the apical region. At the apical region you have what beta? Apical region you have shoot apical meristem. Okay. And if you take a matured leaf, if this is the branch and this is the leaf structure or a branchlet, this region is called as axil. At the axil region over here, axillary bud arises. Shoot apical meristem, axil region, and at the tip you have leaf primordia. Got it beta? Shoot apical meristem, at the tip leaf primordia, and the axil region you have axillary bud. And here in the branchlets, in the leaflets you have differentiating vascular tissue. This is the diagrammatic description of root apical meristem and shoot apical meristem. You are open to ask any doubts? I will give you 1 minute time. You are open to ask any doubts beta. Clear, right? Clear till here? Yeah. Now you see. Apical meristem, shoot apical meristem and root apical meristem. Okay. Shoot apical, root apical meristem. Their key activity is to increase the length of plant. Increase the length of the plant. And intercalary meristem, which serves the same purpose, but it is present in between two nodes in the internodal region. In the internodal region. One more specific activity is this will regenerate the meristematic activity. See, for example, grasses or the members that belong to Poaceae members or grasses, which when they are eaten by herbivores, at the apical regions will be cut off, right? Those apical regions will be regenerated back by the intercalary meristem. Intercalary meristem will be regenerate the apical regions. Okay, they will help in regeneration of apical regions, be it shoot apical meristem or root apical meristem. Clear, right? Intercalary meristems will help to regenerate the apical regions. Okay, mostly shoot apical meristems which are eaten by grazing herbivores. Clear, right? This is about apical and intercalary meristems. Now, secondary meristems are lateral meristems. Secondary or lateral meristems. As we have discussed, we have two types of secondary or lateral meristems. Those are what ma? Vascular cambium and cork cambium. Here you can see in the cortex region you have cork cambium. Okay, with the meristematic activity, few cells are formed towards the epidermis. As a result of the meristematic activity of cork cambium, cells, new cells or fresh cells are formed towards epidermis and cells are formed towards the endodermis. Those cells which are formed towards the epidermis are addressed as cork cells. Those cells which are formed towards the endodermis are addressed as secondary cortex. Secondary cortex. And here we have discussed vascular cambium which is present in the stellar region. If it is in between xylem and phloem, it is addressed as intravesicular cambium. If it is in between xylem and phloem, it is intravesicular cambium. If it is present in between two tissues, that is intervesicular cambium. Intravesicular and intervesicular cambium. So these cambiums, intra and intervesicular cambiums, together they will form a cambial ring in a dicot stem. This cambial ring will form what? The cambial activity will generate again new cells, forming out secondary xylem towards the pith region or the center region. secondary xylem and secondary phloem towards the pericycle. Clear ma? Clear till here? Any doubt anywhere? I hope till here it is clear, right beta? Yeah. Here we go. Now you see next. Permanent tissues. Permanent tissues, we have two types of permanent tissues we have discussed, right? What are they? Simple permanent tissues, and complex permanent tissues. Simple permanent tissues includes what? Parenchyma, collenchyma and sclerenchyma. We will learn two tissues under sclerenchyma. Those are fibers and sclerites. Fine, right? Complex permanent tissues, two complex permanent tissues we are going to learn. One is xylem and the second one is phloem. Xylem we will learn it under in angiosperm if you have to consider. Xylem, we have four tissues of xylem: tracheids, vessels, xylem fibers and xylem parenchyma. Phloem, we have four tissues: sieve tubes, companion cells, phloem fibers and phloem parenchyma. This is a basic framework where you can remember all the types of permanent tissues. First we see the simple permanent tissue, that is parenchyma. What are the general characteristic features of parenchyma? As we have discussed, all the cells are isodiametric in the parenchymatous tissue, and plant body is abundantly composed of parenchymatous tissue. Remember that. Plant body is abundantly composed of what tissue beta? Parenchymatous tissue, and all the cells are isodiametric. Isodiametric means all cells exhibit equal dimensions of length, breadth and height. They can be of any shape, again spherical, oval, round, polygonal or elongated. They can be as any shape. Their walls are made up of thin cellulosic cell walls. Parenchyma will have thin cellulosic cell wall. They may either be closely packed or they have small intercellular spaces. They are closely packed or they have small intercellular spaces. Internally, each cell will have a large central vacuole, that is a characteristic feature, remember. Each parenchymatous cell will have a large central vacuole and cytoplasm is arranged at the periphery. Okay. So, these are the keynotes: isodiametric cells, they can be of any shape, small intercellular spaces can be present, cells will have thin cellulosic cell wall, and then they have large central vacuole where cytoplasm is placed at the peripheral end. These are the key points or key takeaways of simple permanent tissue, that is parenchyma. We have different types of parenchymatous tissue based on the function. If parenchymatous tissue has chlorenchyma in it, has chlorophyll in it or chloroplast, you will see it in palisade parenchyma and spongy parenchyma of a leaf. Okay, palisade and spongy parenchyma. These cells will have what? Chloroplast in it. So, those parenchymatous cells which have chloroplast in it and conduct photosynthesis are known as chlorenchymatous tissues. Okay, they are known as chlorenchyma. Those parenchymatous tissues which have chloroplast in it and conduct photosynthesis are addressed as chlorenchyma. Here you see those parenchymatous cells, mostly they are found in hydrophytes, which has air spaces in it, okay, which has air spaces and they help in buoyancy. These type of parenchymatous cells are present in mostly hydrophytes or aquatic plants. Okay, these cells are called as aerenchyma. Next, those parenchymatous tissues which are specific for storage are considered as storage bodies. Fine, these are different types of parenchymatous tissues. Now you see the functions of parenchyma. Functions of parenchyma includes storage of food. They provide turgidity to the softer parts, Protection, they help in protection as they are composed in epidermis as well. They are present in epidermis and they are good water absorbing in epidermis of root, or root epidermis is also considered as epiblema or rhizodermis. Remember that root epidermis is also known as epiblema or rhizodermis. Root epidermis is also called as rhizodermis or epiblema, as it has good water absorbing capacity, and this is again made up of, root epidermis is made up of what? Parenchyma. Lateral conduction between xylem and phloem is conducted through parenchyma. Photosynthesis function, that is sclerenchyma, just now we have seen, for example, palisade parenchyma and spongy parenchyma, and providing buoyancy in the form of aerenchyma in most of the hydrophytes. These are the functions of what? Parenchymatous tissue. Next we will see collenchyma. Whenever you are learning parenchyma, collenchyma and sclerenchyma, learn the structure and functions comparatively. Look at here, cells may be oval, spherical, polygonal, okay, but they are not isodiametric. Parenchymatous cells are isodiametric. It is found either as homogeneous layer or in patches. Cells have collenchyma, cells of collenchyma are much thickened at the corners. They are composed of what? Cellulose, hemicellulose and pectin. Whereas parenchyma cell wall has only cellulose, thin cellulosic cell wall it has, whereas collenchyma consists of what? Cellulose, hemicellulose and pectin. These cells assimilate food when they contain what? Chloroplast. Okay, again they are considered as sclerenchyma only. Those collenchymatous cells which has chloroplast in it are addressed as collenchyma. Intercellular spaces are totally absent. If you take parenchymatous cells, intercellular spaces are present, but in collenchyma, intercellular spaces are absent. Here you see collenchyma cells may be oval, spherical, polygonal. It is found as what? Homogeneous layer or present as patches. At corners you will see thickenings. These thickenings are due to what depositions beta? Cellulose, hemicellulose and pectin, and they assimilate food and they contain chloroplast. Intercellular spaces are totally absent. This is the structural description of collenchyma, and we have revised it in comparison with parenchyma as well. Next, functions you see. They provide mechanical support to the growing parts of the plant. They provide what? Mechanical support to the growing parts of the plant, such as young stem, petiole and leaf. Okay, they provide mechanical support to the growing parts of the plant such as young stem, petiole and leaf. It also provides flexibility to the organs, allows it for bending. It prevents tearing of the leaf as the cell wall is having thickened ends. It also helps in growth and elongation of the organs. All the functions are very important for collenchyma. Next you see sclerenchyma. This tissue occurs mostly in the hypodermis, vascular regions, cortex, stem and leaves. Hypodermis, you see sclerenchymatous hypodermis is seen in monocot stem, and they are present as xylem fibers and phloem fibers are made up of what? Sclerenchyma only. They are sclerenchymatous tissues. Structure, if you see, sclerenchyma consists of long and narrow cells. They are thick and lignified cell walls. The cell wall is thickened with what? Lignified cell walls. Just compare the cell wall of parenchyma. Parenchyma has thin cellulosic cell wall. Collenchyma has cell wall composition is what beta? Cellulose, hemicellulose and pectin. Here the cell wall is made up of what? Thick and lignified cell wall. Okay, sclerenchymatous. Thick and lignin. Cell wall is composed of lignin in sclerenchyma, and they are usually dead without any protoplast. They do not have any protoplast. They are dead cells. Here if you see fiber, they have narrow lumen and thick cell wall. Scleroid has very narrow lumen and very thick cell walls. You can see the sclerenchyma composition or fibers you can see as xylem fibers and phloem fibers. Next you see the description or the comparative analysis between fibers and scleroids. Fibers you see shape of fibers elongated, thick walled, pointed. In fibers it is elongated, thick walled and pointed. Scleroids, it is spherical, oval and cylindrical. Cell condition, both fibers and scleroids, those are dead cells. Lumen is narrow in fibers, very narrow in sclerites. Occurrence if you see in various parts of the plant like xylem fibers or phloem fibers. Here sclerites if you see, they are scattered in specific parts. Function, they provide mechanical strength. Sclerites, they provide hardness and mechanical support. Uses of fibers, commercial fibers, they are used as commercial fibers as jute, flax and hemp. Sclerites, they are found in nuts of fruit walls, okay, nuts, fruit walls, and pulp of guava, pear and sapota, seed coat of legumes and in tea leaves. This question n number of times asked in your EAPCET and your NEET exam. Where do you find sclerites? Okay, examples include what? Fruit wall of nuts, pulp of guava, pear and sapota, seed coat of legumes and in tea leaves. Next, functions of sclerenchyma if you see, sclerenchyma provides mechanical support to the organs and sclerites will provide strength to the seed coverings. Sclerites form stony endocarp of many fruits in the form of stone fruits, for example almond. Clear, right? Next question. Next you see complex permanent tissues. Complex permanent tissues include what? Vascular bundles, that is xylem and phloem. This is the diagram which is present in the angiosperms. Here you see in xylem you can see tracheids are present, okay, tracheids, next followed by vessels, followed by xylem fibers and xylem parenchyma. This is a complex tissue which has 4 different types of tissues. Phloem you see, at the center you can see sieve tubes and followed by companion cells. Sorry, these are the sieve tubes and these are companion cells. This is what? Sieve tubes which have sieve plates at the peripheral ends and companion cells followed by phloem fibers, lastly phloem parenchyma. Okay, sieve tubes, sorry here sieve tubes, companion cells, phloem fibers and phloem parenchyma. These are complex permanent tissues. Here we go with each tissue description in xylem. First is what? First is tracheids. Okay, tracheids you see. In tracheids, tracheids are elongated tube-like cells. Okay, elongated tube-like cells with pits at the center and they have long tapering ends. Tracheids are elongated tube-like cells with thick and lignified walls, wide lumen is present with long tapering ends. These are dead without protoplasm, dead cells, tracheids are dead cells, they do not have any protoplasm. Thick walls and they have variable thickenings in the inner wall regions. They are present in all vascular plants including gymnosperms. All vascular plants means tracheids are present in all the plants, pteridophytes, gymnosperms and angiosperms. So you can say tracheids are present in all tracheophytes. That is the reason we call them as what? Tracheophytes. Vessels you see. Vessels, they are long cylindrical, okay, they are long cylindrical, they are not elongated like tracheids. Vessels are long and cylindrical, tube-like structures and these are called, one tube has many cells. Here in tracheids, one tube has one cell, but in vessels, one tube-like structure is made up of many cells called as vessel members. They have what? Lignified walls and these are also dead cells without protoplasm. Vessel members are connected to each other. One vessel is connected to other vessel tube by what? Perforation plates. Okay. So tracheids will have pits and pit fields. Vessels will have perforation plates. Amongst tracheids and vessels, which will conduct maximum amount of water, though your answer should be vessels. Vessels will conduct maximum amount of water and minerals. So the vessel cells are devoid of what? Protoplasm. Vessel members have interconnected. They are interconnected by what? Perforations in their common walls. The presence of vessels is characteristic feature of angiosperms. Okay, presence of vessels is what? Characteristic feature of angiosperms. Xylem fibers you see, they have high thickened walls. Dead cells they have thick walls with obliterated lumen. Obliterated lumen means nothing but narrow lumen. Don't get confused with these terms. This terminology will be used in your questionnaire. Obliterated lumen means what ma? Narrow lumen. They can be septate or aseptate, means they can be divided or not divided. The main purpose of xylem fiber is mechanical support. They are again present in all vascular plants. Vessels are absent in gymnosperms. That is a major point to be noted. Vessels are absent in gymnosperms. Next, xylem parenchyma. This is the only living component that is present in the xylem tissues. This is the only living component in the xylem tissues, which is thin walled living cells. The wall is made up of what? Cellulose. It stores starch, fat and tannins. Ray parenchyma, which aids the radial conduction of water. Fine. And this also helps in storage of food and radial conduction. These are present in all vascular plants again. Xylem parenchyma is present in all vascular plants. Vessels are only absent in gymnosperms. Next you see here, exarch and endarch condition of xylem. So, the first formed xylem in the plant tissues is what ma? Primary xylem. Yes or no? Primary xylem. This primary xylem, how it is formed or how it is growing inside a plant, based on that exarch endarch condition is there. For example, if the primitive form of primary xylem is called as primitive form or first formed primary xylem. First formed primary xylem is called as protoxylem. Okay, first formed primary xylem is protoxylem. Fine ma. Mature xylem or later formed primary xylem is called as metaxylem. If xylem maturation is starting from the periphery and going to the center, that is protoxylem is formed at the periphery and maturing to the center, then it is called as what? Exarch condition, which is observed in the root. Protoxylem is towards periphery, metaxylem is towards the pith or center, that is seen in the exarch condition. In endarch condition, if you see, protoxylem, that is primarily formed, first formed primary xylem or primitive xylem will be at the center and it gets matured towards the periphery, that is metaxylem will be towards the periphery, that is endarch type of xylem arrangement in stem. So, the protoxylem and metaxylem or exarch condition or endarch condition is based on the vascular bundle formation or maturation in stem and root of primary xylem, not secondary xylem. Secondary xylem has no role to play to decide whether it is exarch or endarch. Based on the growth of primary xylem, we are saying whether it is exarch condition or endarch condition. If protoxylem is towards the periphery and it is maturing to the center, it is called as exarch condition. If protoxylem is at the center and it is maturing to the periphery, it is called as endarch condition. Okay, clear, right ma? So, here in the exarch what happens? Exarch condition, protoxylem is towards periphery and metaxylem is towards pith or center. Endarch condition, this is seen in where? Roots. Endarch condition which is observed in stem, where protoxylem is towards center or pith, metaxylem is towards periphery. Clear ma? Clear till here? Yeah. Next, phloem you see. It is a complex permanent tissue composed of sieve tube elements, companion cells, phloem parenchyma and phloem fibers. Here the point to be highlighted or noted that in gymnosperms you don't have sieve tubes and companion cells. Rather, in the place of sieve tubes you have sieve cells, and in the place of companion cells you have albuminous cells. Major point, many a times asked in your NEET. First you see sieve tube elements. Sieve tube elements or sieve tube cells, they are enucleated, they do not have any nucleus and they have perforations at the end. Those perforations are called as sieve plates. Okay. Mature cells, they do not have nucleus but they have large vacuole and cytoplasm. The major function is the functions of sieve tube elements. These are sieve tube elements and this is companion cell, right. The function of sieve tube cells are coordinated or regulated by companion cells. Sieve tube cells function is coordinated by what? Companion cells. That is why sieve tube cells and companion cells together called as sister cells. They are together called as sister cells and they are ontogenetically related. They are ontogenetically related. Clear, right ma? This is the picture about sieve tube elements. Next, companion cells, you see, these are specialized living parenchymatous cells. They are closely associated with sieve tubes. They are connected to sieve tubes by a what? Pit fields. These are common in longitudinal walls. Companion cells contain nucleus and dense cytoplasm. They will help in maintaining the pressure gradient of sieve tube elements, like how much it has to conduct the food materials. It will be regulated by what? Companion cells. So, this will maintain the pressure gradient. They will control and support the sieve tube functioning. Lastly, phloem fibers. Phloem fibers are also known as bast fibers. Okay, phloem fibers are also known as bast fibers. They are composed of sclerenchymatous and dead cells. They are long pointed needle like structures. They are long and pointed and they have needle ends. Cell walls is thick and lignified cell walls, you can see. Lignified cell walls, generally they are absent in primary phloem, but phloem fibers are present in the secondary phloem. Mostly phloem fibers are useful. They have a high commercial use in making what? Ropes, sacks, for example jute, flax, hemp. Okay. Which phylum tissue will have commercial use? Then your answer should be phloem fibers or bast fibers. Lastly, phloem parenchyma. Again, this is the living component, not the only living component, because companion cells is also nucleated. So, this is the living, elongated, and they have tapering cylindrical cells. Fine. Cell wall is made up of cellulose here. Phloem parenchyma, cell wall is made up of cellulose and they have pits. They have plasmodesmatal connections. Okay, plasmodesmatal connections in between the cells for intercellular transport. Okay, in between the cells, phloem parenchyma will have what? Connections? Plasmodesmatal connections. They store food material. Food materials are stored in phloem parenchyma like starch, raisins, latex and mucilage. This is the food material which is present in the phloem parenchyma. Phloem parenchyma is absent in most of the monocotyledonous class of angiosperms. In which class phloem parenchyma is absent? Monocotyledonous class. Now, types of phloem. Phloem, again you have two types, exarch and endarch condition again. Exarch and endarch condition. Fine, right ma? Exarch you will see in roots again. In roots you will observe exarch condition. In stem you will see endarch condition. Same like what you observed in xylem, that is exarch and endarch condition. Exarch in roots and endarch in stem. Same the case you will observe for phloem. Here you see exarch condition, where primary phloem will again have primary phloem. The first formed primary phloem or the primitive primary phloem is considered as what? Protophloem. The later formed primary phloem or matured form of primary phloem is what? Metaphloem. Okay. If protophloem is towards periphery and metaphloem is towards the center, then that is called as what condition? Exarch condition that you will see in roots. Same as that of xylem. If protophloem is towards the center, metaphloem is towards the periphery, that you will observe in stem, that is endarch condition. So, this is about types of phloem beta, and then we will see the how vascular bundles are arranged. We have different types of arrangement on which we will use the terms like radial, conjoint, collateral, bicollateral. This we will see in our next class. This along with tissue systems and comparative analysis between dicot, monocot, shoot, root, stem and leaf, we will see in the next class. So, that is all for today's session, students. Thank you. If you are having any doubts or clarifications, you can drop a message in the comment box. I will be addressing all your doubts personally. Thank you.